disconnect or?
JVR, am I audible?
Yes,
madam, you are audible, madam. It was coming in the YouTube also.
Okay. Please, can anyone you share the YouTube to others, please?
Yeah, okay, madam.
Okay, I will allow the participants also.
Yeah, you hello, madam.
Yes, I allowed them.
Yeah.
Good morning, everyone. Myself, Dr. Sharmila Nirojini, Associate Professor, Head of the Department of Pharmacy Practice, Chalpati Institute of Pharmaceutical Sciences, Long Guntur. Welcome you all for the third day of our International Faculty Development Program. I thank each and every participants for your kind support for the last 2 days. Our session, the whole session is being recorded. Those who are facing problem to join with the Zoom, you can watch this program on our YouTube channel. Today we are having here Dr. Narasimhan Guruswamy, Research Associate, Department of Bioscience Research, University of Tennessee Health Science Center, Memphis, USA. It's my privilege to read out the profile of Dr. Narasimhan Guruswamy.
Vinita.
Dr. Narasimhan Guruswamy presently working as a research associate, Department of Bioscience, University of Tennessee Health Science Research Center, Tennessee, United States of America. He has completed his UG and PG in pharmacy in India and has completed his PhD at Niigata University of Pharmaceutical Sciences, Japan. He has completed his postdoctoral fellowship in University of Connecticut, Farmington, USA. He has been associated with the Harvard Medical School, USA as a senior postdoctoral fellow and as well as an instructor. He has also been worked as an assistant professor in King Khalid University, Aba, UAE. His research interests mainly in the area of stem cells, epigenetics, and autophagy, etc. To date, he has also published more than 55 papers in peer-reviewed international journals with 2,763 citations and 6 book chapters. He has given good number of oral and poster presentations in national and international conferences. With this brief introduction, I am welcoming Dr. Narasimhan Guruswamy to deliver his lecture. I am welcoming you, sir. Sir, am I audible to you?
Can you hear me? Is it okay? Can I start?
Yes, sir. Sir, we are audible for your voice. Please share your screen again and you can start the session.
Sure. Okay, sure. Okay. Yeah. So again, thank you very much, Dr. Sharmila, for the introduction. And thank you for inviting me to present my piece of work. Basically, it is a kind of lecture I have prepared because for a faculty development program. And thank you for your institution for this this effort. So today I'm interested to present the topic. It is application of induced pluripotent stem cells. I believe so this will be helpful for the pharmacy students and for the faculties. So it has a lot of pharmaceutical applications. Okay, so so you can just listen to this, and it will be interesting for you definitely. And I will try to make it as simple as possible. And a lot of this pharmacy faculties. You can work on these applications. So my topic again, applications of induced pluripotent stem cells. So I will go by one, like, so from the basic, what are stem cells? Okay, stem cells have the potential to develop into many different cell types. Okay. Basically, it is there in all parts of the body. In every organ, it has a stem cells. Okay. So the main functions of stem cells, okay, is there to repair when there is a damage to the organ. So the basic function, okay, the stem cells are there during the development of embryo. So there it helps for the development for the whole body. After that, we have adult stem cells. So those after the development, after we born, we have the stem cells which help to repair when there's a damage. For example, patient have a heart attack. If when the patient is normal, okay, his heart has the stem cells which can able to go and repair the cardiac tissue where there was a damage. Okay, so this is what we expect. So like that, when there's a damage to the brain, so we have brain stem cells that will go and repair the damaged brain. So it is definitely doing a lot of good things. Okay, so now if you see, there are 3 different types of stem cells are there. Okay, okay, so there are, as I said, adult stem cells. Okay, so which are present in our body almost in every area. Which help to form the embryo and later for the development of the fetal development and the baby. Okay, so that is embryonic stem cells. The third one is induced pluripotent stem cells. That is our main interest today. So before going into that, so what we can call as stem cells? Okay, so we cannot call every cell as a stem cell because stem cell means it should have a specific characteristics. Okay, so it should have at least 3 specific characteristics. So one is self-renewal. That is the main important one. So self-renewal means it should able to renew itself. Okay, and it should be clonogenic. That means it should proliferate itself. Okay, once I will use the PowerPoint and okay. It should self-renew and it should able to proliferate. So that means, for example, as I said, when there's a damage to the cardiac tissue, it should go to the place where there's a damage, and it should able to multiply, because one cell is not enough. So it should able to replicate and that is called clonogenic. Okay, so it should able to proliferate. So self-renewal, that means self-renewal, simply I can say one stem cell should able to form another stem cell. Okay, and also the stem cell characteristics should not be lost during the replication. Okay, that is a characteristic of self-renewal, and it should able to proliferate. And the third important characteristic is the differentiate into specialized cell types. For example, it should able to form myocytes when there is a damage to the heart, okay, cardiac muscles, and also when there is a damage. So of course, the damage can be at the nearby blood vessels, so it should able to form blood vessels also. So it should have the multipotentiality. That means the stem cell should able to convert to not only myocytes, it should also able to convert into smooth muscle cells to form the blood vessels or endothelial cells. So this is called multipotentiality. Okay, so you can call a cell as a stem cell if it has three characteristics. One is self-renewal. Second one, that is clonogenic, or it should able to proliferate, okay, like multiplication. And third one is it should have the multipotential characteristic. That means able to form into different types of cells. Okay. Okay. So this one, there are three types of stem cells. Basically, you can say embryonic stem cells, adult stem cells, induced pluripotent stem cells. Okay, so the first two are is endogenous, which is there in our body. Okay, embryonic stem cells and adult stem cells are endogenous in nature. Induced pluripotent stem cells are not endogenous, that we create that. Okay, that we'll come to that later. So embryonic stem cells, as you may know, okay, which is helping to form for the development of the fetus. Okay, so we call the inner cell mass at the blastocyst stage. Okay, the inner cell mass, okay, it has the embryonic stem cells which are pluripotent in nature. Okay, so I will come to this terminology later. What is pluripotent means? Okay, so I will also explain other terminologies, pluripotent, multipotent, I already explained. Okay, so embryonic stem cells. Okay, so this blastocyst stage we can expect around 3 days after the fertilization. Okay, so this embryonic stem cells, okay, it will be there for maybe 2 weeks after fertilization, from day 3 to maybe 2 weeks around. Okay, so they are pluripotent in nature, and we can use these cells, okay, for the treatment. For example, if the patient have brain damage, for Parkinson patients, we can use these cells, but there are ethical considerations. In order to get this, we need to destroy embryo, which is unethical. Okay, so destroying an embryo, it is unethical, and a lot of restrictions are there, and it is prohibited. Use of these embryonic stem cells are prohibited in United States. Okay, or it is not funded. Okay, then the second one is adult stem cells. Okay, adult stem cells, as I said before, it is there in many organs, like it is there in the bone marrow. Of course, you might hear that it helps to form the blood cells, and some stem cells are there in the heart, in the brain, in the skin. So those are called adult stem cells. So we call them as adult stem cell because after the development, after the baby has born, okay, fetal development, these stem cells are generated, and it is there in the specific tissues. Their main purpose is to regenerate the damaged tissues. Okay, so when there is a damage to the skin stem cells go there and repair the skin tissue. So this is adult stem cells. So we can also use this, and a lot of clinical trials are going on with this. But the problem with this are isolation and characterization are difficult. Okay, it is not easy. Okay, to identify the where it is located and identify and then isolate and characterize the cells are difficult. And the third one is induced pluripotent stem cells. So this one is not endogenous. Okay, the first two are endogenous, that means which are present in our body. This one is not present in our body, we derive it. Okay, so how we can derive this? Okay, so here we take any cells. For example, we take a skin cells, okay, and we convert it into stem cells. Okay, so it simply to say, okay, it works like a magic. Okay, so a skin cell means that is it's a differentiated cell. Okay, and theoretically, okay, every cell in our body have all the DNA or genome which can able to form a whole body. Okay, theoretically we have that, but since the cells are differentiated, it cannot come back. If it is become skin cell, it cannot go back to form a cardiac cell or brain cell. So that is that differentiation cannot go back. Okay, so using this technology, that is induced pluripotent stem cells, we are reversing it back. Okay, so what we are doing, we are taking a skin cell and we are converting back into stem cells. Okay, so it is this technology was developed not very long ago. Okay, so it was developed around 15 years back. Okay, around 2006. So it's a relatively new technology, and it is very simple now. Okay, probably when they to identify this, they might have struggled, but now we have easy method to induce stem cells. So okay, so it is by simply adding some genes. Okay, we can able to convert the skin cells into any stem cells. Okay, okay. So we will go into that later. So induced pluripotent stem cell means they are not endogenous, that is not part of our body, but we are deriving it from any somatic cells. So here I explained what is totipotency and pluripotency and multipotency. Okay, totipotency means, okay, so it is this is the cells, okay, cell status you can expect in the first 2 days of the embryo formation. Okay, so and the pluripotent means thereafter from day 3 to day like 14. Okay, pluripotency. So there is not much difference between totipotency and pluripotency. So totipotency means the stem cell, okay, there are all the cells are the stem cells, they can able to form the whole body, including the embryo and placenta. So pluripotent stem cells means, okay, as I said before, you can get this from the blastocyst, okay, stage of the embryo. And so these blastocyst cells are pluripotent in nature. So the pluripotent cells are cannot form the placenta. That is the only difference between totipotency and pluripotency. Otherwise, it can able to form all the body tissues. Okay, like all around 200 different types of cells it can form from these pluripotent stem cells. And next is multipotent, means it can multipotent means it can able to form many different cell types. For example, it can able to form if I use the example cardiac cells, it can able to form myocytes, it can able to form endothelial cells, it can able to form smooth muscle cells. So one stem cell, it can able to derive into 3 different types. Okay, that is called multipotentiality. Okay, so there is one more term, that is unipotent. So it unipotent means it can able to form only one type of cell. For example, if it is myocyte progenitor, means it can only form myocytes from that stem cells. They are unipotent in nature. Okay. But anyway, those are really not stem cells. Okay. So here I briefly mentioned the important milestones, okay, towards the development of this very novel technology, that is induced pluripotent stem cells. Okay. So as I said, it works like magic. Okay. So and for this, okay, technology, induced pluripotent stem cell development, the Nobel Prize was awarded in the year 2012. Okay, for Yamanaka, Yamanaka, who was the one, okay, in the year 2006 developed this technology. Okay, so very quickly, okay, like within 6 years, you can say, which is really quick. Okay. So then you can understand the importance of this technology. Okay, so within 6 years of developing this technology, okay, so I explain the like in the 1962 was in frog. Okay, so that is the technology was different. There was no IPS, and they used the somatic cell nuclear CNT. And the same technology was used for cloning the sheep. That is in 1997, probably you might hear this dolly sheep. So for that cloning also, the same technology, the SCNT, nuclear transfer. So I have to explain what is SCNT. And in 81, embryonic stem cells, okay, was developed from the mouse. Okay, they were isolated not neurons. And 98, okay, human embryonic stem cells have been isolated from the blastocyst. And 2001, the cell fusion, okay, was developed. So this is like a technology where we use for in vitro fertilization, IVF, cell fusion. Okay. And in 2006, as I said, Shinya Yamanaka, okay, he developed this induced stem cell method. Okay, so here probably with so many genes, and finally he identified that if we add these 4 genes, okay, it's called OSKM. OSKM means the ACT4, SOX2, KLF4, and CMYC. Okay, when we add these 4 genes to a somatic cell, as cell, and wait for genes, okay, it can form a pluripotent state. So, okay. So that's simple. And so this was in 2006. This was done in mouse, and in 2007 they applied this induced pluripotent technology, the same OSKM, okay, in the human. They developed human cells in 2007, and in 2012 the Nobel Prize was awarded. So here, SCNT, that is somatic cell nuclear transfer. So in the egg, they remove the nucleus and add the somatic cell nucleus into the egg, and that will be converted to this. This cell will be converted to pluripotent stage, and the cell fusion, this cells without removing the nucleus, that is cell fusion technology. And this is the third one is for the induced pluripotent stem cells method. So here we take the somatic cell and we add these 4 factors. Okay, this ACT4, SOX2, KLF4, and CMYC transcription factor. When we add these 4 factors, then the cell, somatic cell, will be converted to pluripotent stem cell. It is and the similar, okay, pluripotent stage can also be obtained with the small molecules. There are a lot of works are going on with this method also, like small molecules like So so those are the small molecules. Okay. And in this mass, you will get the so as a pharmacy faculty, just you can be interested in this, like adding small molecules, or you can work with this four factors to develop pluripotent systems. Okay, so development of iPSCS with transcription factors. Okay, we are not working with the small molecules. So use the OSKM factors is called Yamanaka Factors. He was the one developed this method, and it is also called Yamanaka Factors. This was a Japanese scientist who developed and got the Nobel Prize. And these factors are for and cynic. When they add, what they do, when these four factors are added, it slowly converts them which are differentiated cells. Okay, so the differentiated cells slowly go back to the clock. Go back to the differentiated state. Okay, this method is called reprogramming. Okay, reprogramming. So the reprogramming means it goes back to the differentiated just like in person and in the different stage. And ultimately results in the pluripotent stage. Okay. So that means it can able to form any cell types after that. Okay, so for this you have to wait for like 2 weeks nearly. Okay, so it is simple. Just add these four factors and let this development happens. Okay, so after 2 weeks you get this induced pluripotent stem cells. So in the induced pluripotent stem cells, you can able to convert it into a cell that you want. Okay, for example, if you work with that, you can convert this iPSCS cells into cells that you can use. Okay, so you add other factors and you cells. If you work with the heart cell, you add a few factors and it will learn into so here we use these factors, and there are other factors that are also used like and pH. Okay. So this briefly I mentioned characteristics of the somatic cells and iPS cells. So the somatic cells, it cannot it cannot proliferate much. You have to do, but induced iPS cells, because we have transferred the somatic cell into iPS cells. So the iPS cell you can proliferate, okay, for a long time. That's the advantage. It can proliferate. And it has, you can see image, it can be able to transfer it to any cell type. Okay. Okay, so that's the main point. So here, briefly, I said we have so okay, so you can simply add the DNA. So when Dr. Yamanaka was doing, he was using the lentiviral method. He used the lentivirus, okay, and in the lentivirus, he cloned these 4 factors and applied, and he got results. It has advantage that it is like is good, but the problem is the virus, as you know, it can integrate in the genome and it can cause mutation and later. Okay. So that because of the genetic mutation, the virus can produce like retrovirus or lentivirus, so they can cause and another method is adenovirus. You add these factors with the adenovirus. The advantage with this is that this adenovirus do not interfere with the do not integrate with the DNA. And then third method is simply you add the plasmids, okay? Like put it in a plasmid, and then this plasmid will be expressed in the embryonic the somatic cells, then converted to iPSCS. So here I just mentioned what are the cells or cells. The cells has also urine. Okay, so you urine and you can isolate the tubular cells from the urine, isolate these cells, and add factors to get the cells, and you can go for the plasmid transfection. Okay, so here factors and different methods of transfection. So you can, this is urinary method, like there is no any puncturing to collect the blood. There is no you are not damaging any tissues. So this is the colony of induced pluripotent stem cells. Okay, just look carefully. So on the periphery, you can see the differentiated cells, the somatic cells. Okay, the skin, say example, skin cells. Okay, and the center, it is a clone of iPS cells. So the somatic cells converted into iPS cells. These cells are pluripotent in nature, and there is a clear demarcation. So it is called a clone because all those cells, okay, there may be hundreds of cells. Okay, all those cells have derived from one parent cell. Okay, one cell divided, multiplied for a long time, and form a clone. So this is called clone. So the iPS cells have several typical characteristics. So mainly you can say they are called like the embryonic stem cell-like structures, tightly packed colonies with the sharp edges, as you have seen in the previous slide. And it expresses the alkaline phosphatase that these are the pluripotency markers. We want to see this because we want to convert it into pluripotent stem cells. So we expect that these alkaline phosphatase activity is high, and also the other pluripotent genes are expressed. Okay, and this there are a lot of epigenetic changes, and this happening. Maybe if you are interested, we can discuss this later. Okay, so here we have the iPS cells derived from any fibroblast cells. Okay, and now with these iPS cells, as I said before, you can convert it into any type of cells. If you want neuron, just you add some factors on this iPS cell, it will be converted to neural cells. Okay, if you add other factors, okay, it will be converted to cardiomyocytes, like that pancreatic beta cells. Okay, so it depends on your interest. So here are the advantage, like so here it is an example for the formation of cardiomyocytes. Okay, so the somatic cells converted to into iPS cells, the iPS cells, okay, converted converted into the cardiomyocytes. So you can see them. So the same cells, okay, you add a few other factors and wait for 1 week, 10 days time. So it will be forming into cardiomyocytes. Okay. That's why I said it works like magic. Okay. And now what we can do with this? What we can do with this cardiomyocytes? Okay. Either we can go for cell-based therapy. So now you have cardiomyocytes. For example, a patient have a heart attack. Okay. So you take a stem cells from the patient and you convert it to iPS and then into cardiomyocytes. Now you have the patient's cardiomyocytes, you can multiply it, and you can give these cells as a cell therapy for the patient, which can cure the damage of heart tissue. This is our trials are going on. So it is not yet completely finished. It is still trials are going on. And okay, that is one application of this message. Another one, disease model. So you can use this as a disease model, drug free. Okay, we are more interested in this drug screening and toxicological studies. Okay. So this is a slide showing cell therapy. Okay, as I said before.
Sir, Narasimhan sir, excuse me, sir.
Yes.
Sir, sorry for the disturbance. Your voice is not clear, sir. Something disturbance there. I hope that just once you check the net connection,
Okay.
it's something like breaking of voids from past 2-3 slides.
I see. Is it okay now?
Yes, is it okay, sir? Please, if you don't mind, can you go back to 2-3 slides back, please?
This one?
Back, sir. One more, sir.
Here?
One more, sir. Yes,
Here?
sir. Yes, please, sir.
Okay. So here, so based on this, okay, this is the colony of iPS cells, okay. So these iPS cells have the specific characteristics, okay, like they are tightly packed colonies with the sharp edges, and it will express the pluripotency markers, okay. So that means it start to express alkaline phosphatase activity. So this alkaline phosphatase activity is known to be very high in the pluripotent stem cells. Okay, we want to see that. Okay, and also a lot of other genes like ACT4, NONOG. Okay, so these genes are start to be activated. Okay, in these iPS cells. Okay, so these are the indication that our somatic cells has been converted into pluripotent stem cells. Okay, and it's same like other cells. It maintain the diploid karyotype. Okay, and this epigenetic changes during this iPS development. A lot of epigenetic changes are happening. Okay, so this we can discuss later. Okay. Because it's I take more time to explain. Okay. So now we have this iPS cells, okay, developed from the somatic cell by adding these factors. You can convert these iPS cells into any cells as you want. For example, if you work with the pancreatic beta cells, okay, you add a few factors on the iPS cells, it will be converted into pancreatic beta cells. Okay, you can use these cells for your studies. You can use the pancreatic these cells for diabetic patient, type 1 diabetic patients. Okay, you can study, you can test your drug, okay, with these pancreatic beta cells. Okay, and if you are working on the neurons, okay, this you can convert this iPS cells into neurons. Okay, these neurons, okay, you can convert it into dopaminergic neurons. And this dopaminergic neuron, as you know, you can give this to Parkinson patient, and this trial is going on right now. Okay, so the clinical trial is going on. Okay, and you can, if you are working with the cardiomyocytes, you can convert the iPS cells into cardiomyocyte and test your drugs for the or drug effect on the heart. Okay, so that is advantage with this. So this slide shows the example of cardiomyocyte formation from the somatic cells. Okay, the somatic cell which have been converted into iPS cells, and these iPS cells have been converted into cardiomyocytes. Okay, you can see the it can be fibroblast has been converted into cardiomyocyte. It is beautiful, and that's why I said that it was like magic. Okay, so now what you can do with these cardiomyocytes? So you can go for cell therapy. Okay, you can use these cardiomyocytes for a patient with the myocardial infarction, or you can use the cells as a disease modeling. Okay, we will discuss one by one later in the next slides. Okay, and for drug screening and toxicological studies. So this slide explains the cell therapy. Okay, so now you have the iPS cells. Okay, you want to go for treating the patients with heart attack. So you convert these iPS cells into myocytes. Okay, so the myocytes you proliferate and characterize it, and characterized iPS cells can be delivered back to the patient. Okay, and for repairing the heart. The same way you can do for brain damage. Okay, so these are theoretically it is possible now. Okay, not yet it has gone to that stage now with the iPS cells. Okay, theoretically it is possible now. A lot of difficulties are there, we can discuss at the end. So like that, you can do with the somatic cell, you can convert it into any type of cells, and that you can give back to the patient. The great advantage with this is the autologous in nature. So autologous means you are getting from the patient and giving back to the same patient. So there is no possibility for the rejection. Okay, so normally when you take one cell from one patient if you give to another patient, the cell will be rejected. Okay, so here there is no possibility because you are taking from the same patient and giving back to the same patient. Autologous cell therapy. And what are the pharmaceutical applications? So now you have the cardiomyocytes or pancreatic beta cells. Okay, you can test with your drugs. For example, you might have synthesized like 10 drugs or 15 drugs or 500 drugs. You may have a library of 1,000 drugs. Okay, so to test that 1,000 drugs, you need to spend a lot of money. Okay, you need to do a lot of work. But with this, okay, iPS cells or with this iPS derived cells, you can easily screen whether it works or not. Okay, so you can try with the different dose. Okay, if it works, you can go for a different dose. So the library screening and the target identification. Okay, now you have found few drugs are working. Okay, you have 100 drugs, and few drugs are working. It is, for example, it improves the cardiac function. Okay, so now you want to see the mechanism. Okay, how? Okay, so that target identification, okay, you can see, you can study the mechanisms, okay, with these cells. And other important application, the toxicity screening. Okay, as a pharmacist, you may know, we spend like a lot of money for animals, buying the animals and maintaining the animals, and also it is now, okay, we have a lot of restrictions to use the animals. Okay, and in one way, if we use the iPS cells, we don't have to use the animals. Okay, we are saving money, and also ethically we are doing good. So you are expecting cardiac toxicity with your drug. Okay, so you put your drug with the cardiomyocytes, the derived cardiomyocytes. Okay, so you say that derived cells. Okay, if you expect toxicity to the pancreatic cells, you derive the pancreatic cells and test it. Okay, so this for the toxicological applications. Okay, I would say this is the major thing we can do that, like library screening. As a pharmacy faculty or a student, you can just focus on this, like test your drug with the iPS cells, okay, for the efficacy and also for the toxicity. It saves a lot of money. And the disease modeling, okay, so here the disease modeling means, okay, we can develop the disease in the dish. That means a patient have a genetic disease, okay, take the skin cell or blood cell or urine cell from the patient, okay, and we can develop iPS cells and okay, and derive the cardiomyocytes. Now you have the cardiomyocytes which have the same genetic defect as he has in the body. Okay, so that's why it's called a disease model in a dish. Okay, we have a disease model in a dish, and you can try with the new compounds. Okay, so because of this genetic defect, okay, some drugs may not work. Okay, so now you have the disease model, and you can try with your drugs. Okay, so you may have a hit. So this is example for disease modeling. Okay, if you are working on a specific genetic disease, so this iPS method is okay, the main one you can consider, okay, to develop the disease model in a dish. And this it's another application. Okay, still a lot of things need to be done for this. So here mainly we are correcting the disease. Okay, like as I said before, a patient have a genetic disease, and is there a possibility to correct it? For example, he has a point mutation. Okay, as you know, in a sickle cell anemia, there's a point mutation. So now we have some technology. It's called CRISPR-Cas9. Okay, so using the technology, you can correct the gene. Okay, and correct it into a normal gene, and now you have the iPS cells with the normal gene. Okay, put that normal gene into the iPS cell, grow it. Now you have the normal gene from the same patient, and you can go for therapy with this, the corrected gene. Okay, so still it's a long way to go. Okay, it's not it in practice, only in the research. And this is a comparison of this iPS-based drug discovery and conventional drug discovery. As you know, for conventional drug discovery, it may take like years, 10 to 15 years, and it costs a lot of money. Okay, like $1 billion, $1 or $2 billion. Okay, with iPS cells, it greatly reduced the cost because you don't have to go up to the clinical trial 1, 2 to find the cardiotoxicity. Okay, you might hear that several drugs have been withdrawn from the market because at the later stage it was found that it has cardiotoxicity. Mainly it causes arrhythmias. Okay, so after several years and spending a lot of money, it has identified that it has cardiotoxicity. So now you have these cardiomyocytes, okay, from the human patient, and we can do this testing. Okay, we can do this screening at the early stage before going for human trials. Okay, so now you have this opportunity. So you don't have to, if you find something is wrong with this, you don't have to go up to here to the patient. Okay, so now if you have some success with this iPS cells, okay, then you can go for the next stage, okay, for the in vivo and for the human trial. So with this iPS technology, okay, we can go for toxicological screening, and also we can go for drug efficacy. Okay, at least you may have some preliminary idea. Okay. So just to add that, okay, in addition to the drug efficacy, if you are interested in studying the molecular mechanisms and the with the genetic model, you can go for the pathophysiological mechanism, like how this is happening. Okay, so all those things you can study with these iPS cells, which is very difficult with the tissues. Okay, but with iPS cells all easy. Okay, so those are the applications and the challenges. If you see with the iPS cells, it can able to form, if you give, okay, it can form tumors. Okay, it can cause like cancerous tissue, and that's why it is not fully approved. Okay, and if you give, there is a possibility of immune rejection. So when you give, if you take from the same patient and give it to the same patient, there will not be rejection. But from one person to another person, there is a lot of chance for rejection, because still there is a difference in the immunohistocompatibility complex. Okay. And there is also a genomic stability. Okay, so the summary slide, and so for the drug-based therapy, okay, you can use, and also cell-based therapy. So as a pharmacist, we may be more interested in testing our drugs, and also for toxicological screening. Okay, so with this, I conclude, and thank you very much for all of your attention. And
Thank you so much, sir. So now we will give to the participants, if they are having any doubts, they can clarify with you for a few more minutes.
Yeah, sure, yeah.
I request the participants, if you want to interact with sir, please raise your hand. Yes, I find here Dr. Chandrasekhar Budi, he want to ask some doubt.
Sure.
Sir, Dr. Chandrasekhar, just I am unmute you, you can ask your doubt. Yes, Dr. Chandrasekhar.
Good morning, sir. This is Chandrasekhar, sir. Actually, I am a postgraduate student.
Okay.
Yeah, sir. I want to know about this ICP stem cells. Will helpful in these motor disorders also there no, sir. Some motor disabilities were there no, sir.
Yes, definitely. Yeah.
In
definitely,
the treatment
yeah.
also, is this helpful, sir?
Yes, definitely. As I said, a trial is going on for the Parkinson disease. Okay.
Yes, sir. Yes.
So they develop into the dopaminergic neurons. Okay, and these dopaminergic neurons were given to the patient, and the trial is going on, and hopefully it works. And
yes, sir. Thank you, sir. Thank you, sir.
Welcome. Okay.
The participants can raise your hand, thereby we can understand. Okay, in absence of any questions, I would like to conclude the session. First, I would like to thank Dr. Narasimhan sir, for your kind acceptance for being a part of this international FDP program. I'm really thankful to you. We know that you are busy with your research schedules, but even though you spend some time with us to convey these things, really this is a useful session, sir. I hope that every pharmacy teacher and the research aspirant, they got some hope by learning of this induced pluripotent stem cells and their applications, how we are treating the diseases. You mentioned the word diseases on the dish. That is the thing we developed, how much we developed in the pharmacy field. So everyone, they will be this is a good initiation, those who want to do research on this area. Thank you so much, sir, for your kind cooperation and being with us to share your informations relating this induced pluripotent stem cell. Thank you so much, sir.
Thank you, Dr. Sarmila. Okay, thank you very much for the opportunity. And
Thank you, sir. So I thank each and every participant, even though today is Sunday, you have shown your interest. Sorry, we are regretting for our inconvenience during the session due to that audio disturbances. So immediately we rectified it, and kindly sir also accepted to tell, means explain the slides once again. I thank each and every participant who has actively participated here and also watching our channel on YouTube. Tomorrow session, it will be starts at exactly by 10 o'clock. I kindly request you all to join with us, and you will be find the feedback form under the YouTube link. There you can be find the name of our college, Chalpati Institute of Pharmaceutical Sciences. Just by clicking on it, you will be getting the feedback form. Just you fill and forward it to us. At the end of the session, that is tomorrow, you will be received the same way your e-certificate also. I thank each and everyone, and I thank our principal sir and management for giving this opportunity to conducting this type of international FDP program by our department. Thank you everyone. Have a good Sunday. Sir, I'm ending the session. Thank you so much, sir.
Thank you very much. Okay, thank you.